Hi everyone, my name is Lauren Mitchell, and the title of my long talk is Could a Little Bit of a Bad Thing Be Good? And this is in reference to a relatively new idea of hormesis being associated with exposing different organisms to heavy metals, which I will discuss what that means when talking about the background. And pictured below are some biomonitors, which I will discuss also in the background, that will basically be tested for hormesis in the presence of exposure to heavy metals. Here's an outline of the things that I will be discussing, such as the background, which will include those important terms that I mentioned in the previous slide. Also, a research study that supports my topic of hormetic effects of heavy metals. Um, Also, this will include the hypothesis and rationale, significant results, and the next steps that should be taken. Next, the key takeaways will be the final source of my discussion, where I will discuss heavy metals and hormesis, the bad effects of heavy metals and the positive effects of heavy metals, and how this can be a good thing when it comes to the medical field. As for important background that I feel that everyone should know, um, about heavy metals is that they are harmful to living organisms. There has been many research studies that have brought support for this. They're seen as harmful because they cause DNA damage and other negative effects to different processes like reproduction. But how do these actually get into these aquatic ecosystems to affect certain organisms? Well, the answer to this is that human activity and other natural sources cause this. So, a perfect example of this is city sewage systems, which actually deposit runoff into water systems. Um, and many of these contain these heavy metals that we will discuss. And so, cadmium, lead, and zinc are the heavy metals that will be the basis of the research study that will be discussed. Next, biomonitors are very important because they are organisms with structures that are able to accumulate evidence of pollutants in their water ecosystems over time. And this allows researchers to actually study the effects of heavy metals on these organisms and their processes. So this is very important. And some of these organisms that are good biomonitors are aquatic mussels, aquatic snails, and algae. And actually, aquatic snails and algae are the subjects of the actual research study that I will be discussing later. Next, hormesis is very important because um, it deals with the positive effects associated with low doses of toxic substances being exposed to organisms, which could, like I said, they could enhance or stimulate different essential processes like reproduction and growth in these organisms, which this is very interesting because we don't usually hear good things about exposure to heavy metals. Also, hormesis is a particularly new area of research. Um, so there is more field research that is needed for more scientists to adopt this particular area of study, which could be very good for the medical field. Um, and more field research is actually needed because each research study that is done has limitations. But if a larger area of the population can be covered by the information in these research studies, then there could be more support for hormesis. And what I want to drive one's attention to is that in this hormesis diagram, the shape that is made when plotting the amount of heavy metals to the expected response, well, the response that they would have to them, is that there is a kind of upside down U shape, which is an indication of hormesis, which would be very important for what we'll see later. This data table is actually from a research study that looked for the toxicity of heavy metals in the aquatic mussels, which was one of the biomonitors that I discussed. And so the TCS, which is the total comet score, just says that at a 0 that there is a minimal or there is no DNA damage associated with exposing these organisms to heavy metals. However, a total comet score of 400 would be maximum DNA damage associated with exposing these organisms to heavy metals. So here I have drawn an arrow to the reference site. So site 1 is the reference site, and this happens to be the site where there is low to none um, pollution happening of heavy metal contaminants. So it is logical that we should see a smaller amount of a smaller total comet score for the organisms here. However, sites 2 through 4 show higher total comet scores, which is an indication that these polluted sites are actually they actually present more DNA damage to the mussels in this study because of their exposure to heavy metals. And these are actually, although these total comet scores don't quite reach 400, which I say is the maximum amount of DNA damage, they do show that there is significant DNA damage associated with the exposure of these organisms to heavy metal concentrations. So this slide is just to say that, just to emphasize the fact that heavy metals are often seen as toxic to many organisms. And now I will be discussing my research study, which, well, the research study that I focused on, which is entitled Hormetic Effects of Heavy Metals in Aquatic Snails, is A Little Bit of Pollution Good, and this is by Hugh Lefcourt and his associates. So let's get right into it. Now we'll be discussing the hypothesis and the rationale concerning the major research study that I will be discussing. And the hypothesis is that the P. columbiana, which were these snails that were from the polluted site, they will be the most likely to exhibit hormetic effects um, when they are exposed to the different heavy metals. And also, the rationale is that they want to achieve, the researchers want to achieve an improved research design because their previous research actually brought support for hormetic effects in these aquatic snails. However, they wanted to refine their design so that they will have more accurate results and better support for the idea of hormesis. So the experimental methods that they use are collecting the snails from polluted and reference sites. And as I mentioned, the species of snail collected from the polluted sites was P. columbiana, and the species collected from the reference sites is L. palustris. And they were transported to Gonzaga University, where these researchers observed them and actually nourished them by feeding them lettuce for 2 weeks first. And this allowed for any weak links to die off, which were these snails that could not handle the stress of a new environment. And also, this allowed for their digestive tracts to be cleared of any heavy metals from their original um, ecosystem. After this, these scientists created many ecosystems for the snails and algae, where they actually took 64 plastic containers, filled them with well water, leaf litter also, and they small amounts of leaf litter just to make the ecosystem seem more normal. And they also um, added in algae in order to gain info about its bout with heavy metals as well. Um, and so after doing so, they let the algae grow for 2 weeks, and then they added 12 of the different 12 of the snails. Um, well, they added 12 snails to each container, and they separated the snails from the algae using a separator, and they also suspended a ceramic um, tile from the top of the container in order for the algae to grow on. And basically, this took away the bias introduced by the snails um, being able to graze on the algae, which would have been bad. So they managed these mini ecosystems for 75 days, which included um, going to the site, the natural site between the two sites that they collected the snails from. And actually, this is where these mini ecosystems were. And they actually added well water when need be each week, because since this was a natural site, evaporation occurred. But the conditions of these mini ecosystems were pretty normal. Also, after these 75 days were up, these snails were actually nourished again to rid their digestive tracts of any heavy metals. Um, and also, the algae and the shells of these snails were actually processed in preparation for them to be analyzed for these concentrations of heavy metals and their effects. So the way that they analyzed these was through emission spectrometry and atomic absorption, which are two different ways that are involved in actually identifying the heavy metals and their concentrations. Um, and also, StatSoft statistical testing was used for analysis of the results to see which findings were statistically significant um, in dealing with the effects of the exposure of heavy metals to these organisms. And so growth and reproduction were the two areas that were actually the very most important things. So this is just a schematic drawing of the experimental design, and I want to bring attention to the fact that there were 4 treatments, none, low, medium, and high, that were in the different tubs. So a treatment of none would mean that they did not put any polluted soil from the polluted site into the um, ecosystem. So this treatment was used as a control. However, as can be inferred, the low treatment was a low amount of the polluted soil being added into the ecosystem, which in this case was 2 milliliters. Um, and then the medium treatment was 16 milliliters of polluted soil, and the high treatment was actually 60 milliliters of polluted soil. And this polluted soil contained lead, zinc, and cadmium, which were the most important heavy metals. Um, and so these were the different, this was how the different mini ecosystems were separated, and then they were analyzed for reproduction and growth. So now I will be presenting the results associated with the snails when they were exposed to these heavy metals. And I have provided a key as to what we are viewing at the very top up here. And so this row comes from the L. palustris, and this row comes from the P. columbiana. And so in figure 1B, what I want to bring everyone's attention to is that the numbers for the amount of lead that was acquired into the structure of the L. palustris snails, the range of these numbers is actually higher, showing that they acquired more of lead than the other heavy metals that they were exposed to. And here I just wanted to emphasize that although these species of snails, the L. palustris and the P. columbiana, were exposed to the same amounts of these heavy metals in the beginning, that they have the ability to be able to acquire some heavy metals more than others, basically. And then I want to bring everyone's attention to this red rectangle around 3A and 3B. And so for 3A, on the x-axis, we see the amount of polluted soil that was added, which is the source of the heavy metals. And on this left y-axis is the mass, which will be a representative of the growth seen in the snails when they were exposed to these heavy metals. And so this brings me to look at the triangles, which represent the mean mass. And this is the overall shape that this data gives. And this shows that when this species of snail, the L. palustris, is exposed to the heavy metals, that they have an overall mostly negative effect on the growth. And so this is not hormesis. Also on the right hand side, on the y-axis is the recruitment abundance, which would be talking about the reproduction seen in these toad populations. And so these boxes, these little squares, show this particular thing, and this is the overall shape of this data, which shows an overall decrease in the reproduction of the L. palustris snails when exposed to increasingly higher doses of these heavy metals. So they do not experience hormesis at all. Whereas in figure 3B, as exhibited by the squares, the P. columbiana actually exhibited hormetic effects in reference to their reproduction, which is indicated by this U shape that I explained that you should see earlier when if hormetic effects are involved. So that's a plus for their reproduction at low doses. And as exhibited by these triangles, they also represented hormetic effects for the growth of these P. columbiana snails when they were exposed to low doses of heavy metals. So the main result here is that the P. columbiana exhibited hormetic effects for all three heavy metals, and the L. palustris did not. So the next thing I want to discuss are the results associated with the algae. And what I want to bring everyone's attention to is that there are no hormetic effects associated with exposing the algae to heavy metals, the oscillatoria algae. And so here, what is shown is that across the polluted and the reference site, the controls produce the most growth in the algae. Um, and what is also shown here is that across the polluted and the reference site is that the high doses of heavy metals actually brought the second highest growth of the algae, which is very unexpected, and which is actually the basis of why no hormetic effects are associated with this. And this could actually stem from the fact that during the low and medium doses, there was actually some water mold fungus that was seen on the ceramic tiles with the algae. And actually this water mold showed hormetic effects, and it actually dominated over the algae in these two treatments, which could be the basis of this particular result from the algae. So here I want to discuss the conclusions and further research associated with the research study. And the most important point was that exposure of organisms to low doses of toxins like heavy metals could be stimulatory to their different processes like reproduction and growth and other processes that they have. And this was seen by the results of the P. columbiana. Conversely, these high doses of these toxins could prove to be very bad for these organisms, which is usually the thing that is seen when discussing the exposure of heavy metals to organisms. That's the more normalized research. And there are limitations associated with this study that say that basically, this research can only be used to draw conclusions about these particular species that were examined and none others. So, if more research is taken for like larger populations and ecosystems, then more mechanistic research, then Hormesis could be further supported, and other scientists will be willing to adopt this particular viewpoint. So on this slide, I just wanted to put some key takeaways. So like, why does everyone care about this particular long talk in hormesis? Well, this says that chemicals thought of as toxic may actually have hormetic effects on certain organisms, which could potentially lead to a breakthrough in the medical field where more toxic substances or stressors in low doses could actually be tested on humans in order to see if they will produce the same hormetic effects to stimulate things like reproduction, growth, or other processes associated with different illnesses. So this could be really put to really good use for humans. Also, another thing is that heavy metals are often seen as toxic due to the DNA damage and other kind of damage that they cause to the genome of different organisms. However, they can also be seen as stimulatory, and this is the biggest thing because most people do not see them as a good thing. So this shows the other perspective of heavy metals in reference to hormesis. And the last key takeaway is that biomonitors are very important. Without biomonitors, important breakthroughs in science couldn't happen, such as these regarding the effect of heavy metals um, on different organisms, because they actually provide a structure that can accumulate these heavy metals and further be analyzed to see the effects, whether harmful or hermetic. Here are my references. Thank you for watching, everyone. Bye.